Electrophoresis is a technique by which different biomolecules can be separated when subjected to an electric field. For its correct performance, many aspects must be taken into account, such as the biomolecule that will be analyzed, the gel concentration, the buffer, the voltage, and so on. In this practice, the aim was to acquire the knowledge and experimental practice to be able to visualize and analyze different samples of nucleic acids and to determine their quality. Also, to decide which concentration of agarose is needed for the different samples, by testing many samples at a time with different concentrations of agarose in two buffers, Chi and TBE. Electrophoresis is a fundamental technique, and it is used very frequently in research laboratories. One of the most used applications is the PCR amplification confirmation, to identify fragments of DNA. It also can be used to determine paternity, identify damaged genes, identify some genetic diseases, check the purity of an antibiotic or vaccine, and so on. For the preparation of an electrophoresis, many aspects must be taken into account, such as those shown in Table 1. About the agarose, you must wait until the gel is completely dry, otherwise the results will be erroneous. If you're in a hurry, you can put it in the refrigerator for 10 to 15 minutes. The concentration of the gel is also important, since in it depends which fragments can be better observed. For example, in a not concentrated gel, bigger fragments will be separated, when in a very concentrated gel, smaller fragments will separate. To be able to validate these results, it is necessary to use a molecular-based peer marker, since it indicates the size of your fragment, and thus it allows you to compare with fragments of interest. The loading buffer adds dye to the sample so it can be observed, and helps increase the density so that the sample does not get diluted in the running buffer. The dyeing agent must be chosen according to the size of the samples to be run. Another important aspect is the direction of the electrophoresis. Since the DNA has negative charge, it will run to the positive side, so the wells must be charged on the negative side, which is usually the black wire. The time of the electrophoresis depends on the size of the sample and the concentration of agarose. To visualize how the band is running in the gel and know when to stop it, it is recommended to use colorants, such as God tag, that have a specific size and can be visualized while the gel is running. But last, the voltage must be calculated according to the distance between the two electrodes. An approximate voltage ensures the samples will correctly run, and that the agarose will maintain its integrity without overheating.
There are two main considerations to take into account when running a sample from DNA or RNA. The first one is the structure. Since DNA has a double-stranded structure, the gel can be run in native conditions. However, since RNA has a single-stranded structure, it must be denatured first to prevent it from forming secondary structures with itself. This would interfere with the results and we wouldn't be able to see the real size of the sample. The second one is the way the bands are shown. For a DNA sample, we would expect to see one band corresponding to the size of the fragment. However, for an RNA sample, we see two bands corresponding to the 28S and 18S subunits of the ribosomal RNA. Sometimes we can even see a third band corresponding to the 5S ribosomal subunit. It's a technique that allows for the visualization of larger fragments of DNA, such as genomes. Contrary to a traditional electrophoresis, in this case the electric field is not kept constant. Instead, the current is delivered at timed intervals from different angles. This prevents the DNA fragments from diffusing because of the change in the electric field that acts upon them. This gives a better resolution for large fragments. The main application for pulsed field gel electrophoresis is genotyping of bacteria, where we want to identify a specific strain of a disease-causing bacteria by analyzing its entire genome. For some solutions, it's important to use water that has not been distilled but that has a molecular biology grade, such as milk water. This is the case for solutions such as the loading buffer, which will come in contact with nucleic acids to avoid its degradation due to the presence of nucleases or salts in the water. It's also important to mention that the loading buffer, as well as the SDS solution, cannot be sterilized because of its components. This is also the case for antibiotics in culture media, which should be added after sterilization to avoid the degradation of its components. In the case of the sock medium, it's important to prepare the salt solutions and the glucose solutions as stocks separately and add them after the medium has been sterilized in the autoclave to prevent degradation of any of these components. It's also necessary to take into account that some solutions must be prepared in a specific order. This is the case of the TBE or TAE buffers, where the first component to be added is the tris, then the EDTA, and finally the acid. This is because EDTA can only solubilize when the solution has a basic pH.
Methodology to do an electrophoresis of nucleic acids in agarose gels. As in the other practice, it is necessary to calculate the amount of material that is going to be needed. In this case, the calculation for the buffer solutions TBE1X and type 1X, and agarose gels at 0.8%, 1.6%, and 2.5%. Once the solutions are ready, wait until the agarose cools down to add the gel red and mix it. Defrost DNA and label each well with the content as well as the order of the different samples. In a piece of paraffin, add drops of loading buffer separated from each other, and with a micropipe, take a sample and mix it with the loading buffer. Empty the mixture in the well that corresponds. Run the samples in the electrophoresis chamber to 60 minutes at 100 volts. Visualize the gel in the transilluminator and keep the record of the image.
Figure 1 shows the patterns obtained from the run of the agarose gels with TAE buffer at different concentrations of agarose, 0.8 for A, 1.26 for B, and 2.5 for C. The first 3 wells in all the gels were loaded with DNA markers, which are helpful to identify DNA or RNA sizes. The fourth well contains a PCR plasmid of topoisomerase. The fifth well was loaded with E. coli genomic DNA. The sixth well was loaded with a sample of Tlaquache's DNA, and the seventh well was loaded with ribosomal 16S PCR product. Figure 2, on the other hand, shows the patterns obtained from the run of agarose gels with TBE buffer at different concentrations of agarose, 0.25 for A, 1.6 for B, and 2.5 for C. The first 3 wells of every gel were loaded with DNA markers. Meanwhile, wells 4 to 7 were loaded as follows. The fourth well contains a PCR plasmid of topoisomerase. The fifth well was loaded with E. coli genomic DNA. The sixth well was loaded with a sample of Tlaquache's DNA, and the seventh was loaded with a ribosomal 16S PCR product.
Figure 3. Identify problems in the samples loaded in the agarose gels with type 1X. A. Agarose gel 0.8%. B. Agarose gel 1.6%. C. Agarose gel 2.5%. Yellow squares correspond to protein contamination in the samples. Blue squares show genetic material degradation, red squares show RNA presence, green squares show excess of silencianol in the loading buffer, and the circles show bubbles of lint present in the gel when photographic.
After checking the results, we can see that the best results are obtained when we use buffer type because it is used for common analysis of DNA and RNA. Also because TBE buffer has a bigger concentration of salt, so it affects the running of the samples. The migration patterns seen in the gels at different percentage of agarose show that it clearly affects the samples because they are a bigger size. So for bigger size of samples, we need to use less percentage of agarose. That's why for these kind of samples, we will be using 8% agarose gels.
To improve our gel preparation skills, we need to take into consideration all the details previously mentioned, from the percentage of agarose, making the calculations, manipulating correctly the samples and the material, among others. The technique used to load the wells was the correct one because we have like a correct migration and gel was punched. On the other hand, we need to be careful with the amount of sample used because we put like more amount of the sample, we're not going to have defined abundance. To avoid the presence of lint in the material, we need to check that it is clean, and if it needs to be washed, we can't dry it with paper towels. This practice was very useful to understand the procedure of making an electrophoresis and to see the errors we can make. And it will help us to correctly choose all the conditions of the electrophoresis that we're going to make and other practices. Is another variable to take into consideration when running an electrophoresis. A higher values of this variable, we're going to have a faster DNA migration, as well as a high voltage will increase the temperature of the buffer so the gel can melt. We're also going to have like poor resolution of the DNA bands and the equipment can be damaged. So it's recommended to use like between 5 and 8 volts per centimeter. On the other hand, if we use low values of voltage, we're going to have scattered bands and the running time will be like way too big. So in this practice we have like the voltage and the running time were like the correct because the samples finished running the gel without going out and it wasn't poured.
The gels with less percentage of agarose allows the samples to migrate faster, although it does not separate well the large fragments. For the 3 concentrations of agarose, the fast DNA marker was the best for both buffers, and the images were better taken with the buffer TBE. The sample of the plasmid had a size of approximately 5 kilobases, and the 3 isoforms were better identified in the gel of 0.8% of agarose. Both the DNA and the RNA were degraded, and additionally the DNA showed a lot of RNA contamination. The quality of the PCR products is good, and they are an expected size of 450 base pairs, and the best volume to produce a short band of it, so it could be analyzed, was 1 microliter. It must be taken into account that the higher the concentration of the agarose, the slower the samples will run, so it is necessary to increase the time.
Finally, some safety precautions must be taken when handling components. For example, SDS is very irritable and tends to form dust, so it should be handled correctly. In the case of highly concentrated acid solutions, such as the ones used for pH adjustment, they should be measured and handled in an extraction hood. This is to avoid aspirating any of the harmful fumes. This is also the case for other toxic components such as phenol.